So now let's proceed to the structure and the function of proteins. Okay, so the first thing that you have to know is that the function of a protein greatly depends on the structure. Okay, so these photos, these figures here are examples of proteins. So hemoglobin, pepsin, and collagen are all proteins. Why is this not working? So it has a protein. As you can see, it is three-dimensional. Okay, so meaning sa three-dimensional, it has an X, Y, and Z na dimension. It's twisted, folded, and coiled into a unique shape. So imagine ang this folding here, for example, this folding here is specific for hemoglobin. So if that folding has changed, then the function will be different. Okay, so it won't function as hemoglobin anymore. Okay, so as I've said, the function highly depends on the structure. Okay, so because of the complicated structure, the complex structure of our proteins, we try to break them down piece by piece to deduce the structure. Okay, so mo nang gitagaan nato og levels of structure ang protein. So ang primary structure sa protein class, ang primary level is the order of amino acids. So all proteins, of course, should have a primary level of the primary structure. Okay, so it's the order of amino acids in chain. So as you know, natay 20 ka amino acids, di ba? So 20 common amino acids katong alanine, valine, kato sila. Okay, so kung unsa ang order sa amino acids, that is the primary structure. So a protein is a polymer of amino acids. Okay, so meaning if you have an amino acid sequence like alanine, if you have like, for example, alanine, valine, glutamic acid, okay, this is the order, this is the primary structure of protein, kung unsa ang pagkasunod-sunod sa ilahang amino acid. So for example, this one, so have lysine, valine, phenylalanine, glycine. If you change that amino acid, if you change like for this one, for example, then that will become a different protein. Okay, so that's the primary structure. Now if there is a slight change in amino acid sequence, like for example this methionine has been interchanged with this alanine, then it could affect the protein structure and the function, because as you know, naaman ni sila, for example, ang methionine, it has a sulfur that can form disulfide bonds, di ba? And then you have a serine here, so this could form SH man ang serine, nasa na S, so it could form disulfide bonds. If ang kining methionine nabalhin pa ni siya diri sa kining space sa alanine, then ari diri ang disulfide bond mo form, di ba? So that would change in the possibly ang pag-curve sa protein, di ba? So imagine a small, even just a single change in amino acid, then that could already disrupt or change the protein structure, and so it could change its function. So one amino acid could make a difference. So for example, in people with sickle cell anemia, it's just a change from glutamic acid, it's a change to valine. So normally ang normal red blood cells, mao ni ang primary structure sa normal nga hemoglobin. But in people with sickle cell anemia, mo nang na-replace ang glutamic acid with valine. Okay, and so that's why the cells look like this. Instead of this one. So in ani ang now. That's why it's called sickle cell, because the cells looks like a sickle. Now, after na to ma-form ang primary structure, let's proceed to the secondary structure. So ang secondary structure is mo ni ang first nga pag-fold sa protein. Okay? So this is the local folding. So sa secondary structure class, these are folding along short sections of polypeptide. Okay? So the short sections ra. So mag-fold ra ni sila. Ang interaction is only between adjacent amino acids. Adjacent meaning tapad ra, dugol ra kaayo, di ba? So for example, in this one, this is the carbonyl portion, di ba? The carbonyl and the amino portion. And then again, this is the carbonyl and the amino portion. So these are just the amino acids, di ba? So ang kini nga mga amino acids class, these are interacting. So from first, second, third. So then the fourth amino acid, for example, this one, the fourth. So ang fourth ug ang first na interaction, di ba? So ang unsa ra ang dugol nila nga amino acid. So this is held together by hydrogen bonding, and the hydrogen bonding is between the O and H of the backbone. Okay? So this one sa NH, di ba? Nya sa carbonyl. So, di ba, we have an amino group o carboxylic acid group. Sa amino acid, we have this one and this one, di ba? So, ang H sa usa ka amino acid will interact with the carbonyl nga oxygen with another amino acid. So mo na sila magka-H bonding silang duha. This will form an H bonding. So mo nang kung unsa ang duol niya, mao na iyang ma-H bonding. That's why it's only between adjacent amino acids. And so don't forget nga sa secondary structure, it is the H bonding between backbones. Okay? Because we also have H bonding sa side chains, di ba? So this is the H bonding between backbones. Okay, so meaning sa backbone class, it's the amino group o sa carbonyl group. Okay, so that's why sa secondary structure class, it could form two structures. It could either be alpha helix or beta pleated sheet. So ang alpha helix is this one. So mao ni ang ma-form niya. Because of that hydrogen bonding, maka-form siya og ingon ani. This is what we call as the alpha helix. Another nga structure nga ma-form due to the H bonding of the backbones, this is still a secondary structure, is called a beta pleated sheet. So in ani siya og noun. Okay, so this H bonding, so this is another polypeptide here. So this H bonding, because of this H bonding, then it could form like a sheet. So that's why it's called a beta pleated sheet, because mo form siya og murag sheet iyang formation. Okay, so for fibrous proteins, you only have secondary structure. So example of that are keratin and silk. Okay, so we will go to that later when we have the types of proteins. So as you can see, this is the alpha helix. So mo form siya og coil, di ba? This due to the H bonding. So mao ni ang mga H bonding sa iyang adjacent nga kanang adjacent niya nga amino acid. Okay, so another thing is the beta pleated sheet. Okay, so di ba inana ang H bonding sa beta pleated sheet. Okay, so aside from that, you have this electrostatic attraction, of course, because you have amino or carboxyl group. This is a positively charged or negatively charged. So that's why there is an electrostatic attraction. Electrostatic meaning it's an attraction between opposite charges. You also have hydrophobic attraction because, for example, you have a valine here. Yang R group sa valine is just CH, CH3, CH3, diba? So sa valine, you have this one. So this is hydrophobic. And if you have here isoleucine, then for isoleucine, inana ang ihang So sa isoleucine, it's like that, diba? So if you have an isoleucine o valine, so both of them are hydrophobic. That's why you also have hydrophobic attraction between the side chains. And then you have metal ion coordination, which we will go later. So like hemoglobin, for example, which has magnesium, diba? Sorry, iron. So what happens is that this metal goes to the center, but we will go to that later for when we reach some other examples. Okay, so that means there are other attractions, but in secondary structure, these are what the hydrogen bonding between the backbones are have formed alpha helix and beta pleated sheets. So that is the secondary structure of the proteins. So next we have the tertiary structure. So this is the whole molecule folding. Okay? So meaning tanan nga attraction included ane. So it is created when the secondary structure fold and form bonds to stabilize the structure into a unique shape. So if you have this one until the secondary structure, if you have that one, and it will have to form another of that, another, and then like you have an, so that is the tertiary structure. Kung unsa na ang lain pagyud nga attraction, it's not just the hydrogen bonding, but it all includes other bonding as well. So it is determined by interaction between R groups. So as you see, apil ng R groups, it's not just the backbone, it's not just the carbonyl and the amino group hydrogen bond which are H bonded to each other, but it includes the R groups. So it includes the hydrophobic interactions, which I showed earlier from the previous slide, and also the disulfide bridges. So disulfide bridges occurs on the amino acids that contain sulfurs. Okay, so that's disulfide bridges. And then you have ionic bonds between R groups. So some R groups have charge, right? So some R groups have like glutamic acid, it's COO minus, and you also have basic amino groups, diba? So they could also form ionic bonds. And then you have hydrogen bonds between backbones. So that is included in the tertiary structure. So the tertiary structure could not form without the secondary structure, diba? So this secondary structure is formed. I mean, this secondary structure is formed from the H bonding of the backbone, and this secondary structure is included in the tertiary structure. That is why ang term na to class is levels of structure. They are not called types of structure but levels, because primary level, it's just the sequence of the amino acids. Secondary level, it's this the folding due to the hydrogen bonding between backbones. And the tertiary is the complete molecule folding. So apil na ang folding due to the side chain, due to the backbone, tanan nga interactions. Okay, that's all the interactions within that polypeptide. So kini class, this is one polypeptide. Okay, so kini tanan sumpot ni these are all connected by peptide bonds. Okay, so you also have the van der Waals. So that's why you form globular proteins. So globular proteins have tertiary structure, and mostly ang globular proteins, they are the enzymes. So mao na ang mga enzymes usually are globular, meaning globular, it's like this kanang ingon ani ang structure. Kay di ba, you have, as I mentioned earlier, you have keratin and silk. These are fibrous proteins. So ang fibrous proteins, they only have up to secondary structure. So mo nang some proteins are just kultubra sila sa ingon ani nga structure, they are plain. So most of them are structural nga proteins. So for example, we have keratin and silk. So these are present in hair, di ba? So mao nang kanang para pampakulot sa hair, para sa perm o para pud pang rebond. So that has something to do with these fibrous proteins. Okay, so that means not all proteins have tertiary structure because we have fibrous proteins, diba? And so we have the last level of structure of protein. So this is what we call as the quaternary. Take note that not all of the proteins, have quaternary. Okay, so some proteins only have up to secondary structure. So they do not have any like side chain interaction. So they just have those H bonds among the H bonds between the backbones, and also a little bit of katong sa disulfide. So kini ba, a little bit of this, so present po ni sila, okay, but it does not contribute a lot, but it still contributes because structure affects the function, diba? So this still contributes, but what forms the secondary structure is this, the hydrogen bonding between backbones. Okay, so in quaternary it's two or more tertiary folded peptide subunits bonded together to make a functional protein. So that means after this tertiary, so you have this tertiary, for example this tertiary, so kini, so you have this tertiary. So again sa tertiary apil ang hydrogen bond. So this is from side chain, serine, gikan sa serine nga side chain, and then gikan sa carboxylic acid nga side chain. Okay, and then we have disulfide bridges, then we have ionic bonds. Okay, so that's included in the tertiary structure. So that's an interaction, additional interaction between the side chains. Okay, so in quaternary structure class is that you have more of this. You don't have just one tertiary structured protein. You don't just have one. It's more than one, and then those proteins are interacting with one another. So it's two or more tertiary folded protein peptide subunits. So such as this one. So kini nga structure, it's just a single nga protein. It's a single polypeptide nga naay primary, secondary, and tertiary structure, di ba? This one is another polypeptide that has primary, secondary, and tertiary structure. Kaya naman siya 3D, di ba? This one is another nga na primary, secondary, tertiary, and so is this one. So when these four interact with one another, they now have what we call as the quaternary level of structure. Okay, and usually mao ni sila ang naay mga iron, mga metals. So katong ganiha naay metal nga interaction. So this one, metal ion coordination. So usually it exists in the quaternary structure. Okay? So for example, we have hemoglobin. So this is the structure of hemoglobin. It has 2 alpha and 2 beta subunits. So mana ang structure sa hemoglobin. And then we have collagen. So collagen is composed of 3 polypeptides. So di ba? Tulo ka buok nga polypeptides, di ba? So it has tertiary. So now you have 3 polypeptides. So meaning this polypeptide is not connected to this via peptide bond. So dili siya connected via peptide bond. So dili connected, meaning dili connected ang ilahang amino acid ends. Okay? So these are 3 separate polypeptides. Okay? And then once nag-interact na ang 3 separate polypeptides, then you have a quaternary structure. Okay, so that's more than 1 polypeptide. Okay, so again, for the primary structure, it's the arrangement of these. So the arrangement of the amino acids, and then for secondary, it could either form alpha helix or beta pleated sheets due to the hydrogen bonding between the backbones. And then once you have additional interactions among the side chains. So for example, you have this one. Now the protein now has a tertiary structure. So for example, this one naa siya ani, naa siya sa beta pleated sheets. So di ba kini siya naka-form og beta pleated sheets. And then napagdaghang daghan pagid siyang liko-liko, di ba? So daghan siyang folding. So that's now have it now contains tertiary structure. And then once this one, so this is one polypeptide. So if it interacts with another polypeptide, so dara ang usa ka polypeptide, so it's this one. So if it interacts with another polypeptide, then that is now it now contains a quaternary structure. Okay, so I hope you understand. Okay, so as you know, proteins are easily denatured. So denaturing a protein means that we are unfolding its structure. So gikan sa mao ni ang functional nga structure sa protein. So mao ni iyang structure jud, it has primary, secondary. So this is a tertiary protein, di ba? It has a tertiary structure. Wala siya quaternary because it's just a single polypeptide. So to denature that means that we are removing the interactions. We are trying to break the interactions. So now we have just a single, so primary level of structure. So it's just the arrangement of the amino acids. Okay? So we are trying to disrupt the tertiary structure. So in order to disrupt a tertiary structure, so to disrupt the disulfide bonds, to disrupt the tertiary, the hydrophobic interactions, the electrostatic interactions, we can change the pH and temperature. So that is why a lot of proteins, a lot of enzymes are sensitive to pH and temperature, because once they experience a change in pH or a change in temperature, then the bonds that held together its structure will break. Okay, so it unravels or denatures a protein. So that it means unfolding a protein means the disruption of H bonds, the ionic bonds, and disulfide bridges. Okay, so it destroys the functionality. Now let's proceed to the classification of proteins. So we can classify proteins based on their structure, their function, and their size. So based on structure, we can classify that as globular, filamentous, or helical. So globular katong ingon ana, di ba? Usually this has the tertiary level. Okay, so mao nang nay mga tertiary level. We have filamentous, so from filament, so di ba mga inana lang filament. Okay, so it could either have tertiary or it could just have this cotoblast secondary, di ba? And we have helical, so we have helical nga proteins. And then we could also group the proteins in terms of function. So in terms of function, they could either be catalytic or they could be structural. So catalytic, these are mainly, most of the proteins are catalytic, classified as catalytic because they act as catalysts. So mao na ang gitawag natong enzymes. Those are the biological catalysts or the substances that speeds up a reaction. And we have structural proteins. So as you know, like collagen, you have keratin. So those are structural proteins. Usually, ang structural proteins are not globular. So usually, they are just filamentous or helical. So ang usual jud nga globular are the catalytic. Okay? And we have small versus large versus very large. So it depends on the protein. So actually, like for example, hemoglobin, it's a very large protein, diba? Because it contains four long polypeptides. So in ana with nadira ang iron. That's iron 2 plus, I think. So you can classify protein also as small, large, and very large. So it depends kung unsa kadaghan ang iyang amino acid. Okay? Now, another terms to describe a structure of a protein or to deduce the structure of a protein are the subunits and the domains. So a single polypeptide may have regions along its length that fold up separately, and those are called domains. So domains naara na siya sa single polypeptide. Okay, so for example in this one, so this is one single polypeptide, diba? This is one single polypeptide, and so this is one domain. This is just one folding. Okay, and another thing is that for more than one polypeptide coming together to form a protein, it's called a subunit. So, for example, sa hemoglobin nga structure, this one is called one subunit. This one is another subunit, and this one is another subunit, another subunit. Okay, that is what we call as a subunit. So, ang subunit is in quaternary proteins. Kay naman siya more than one polypeptide. Okay? We have protein families. So, ang protein families class, these are proteins that have similarities in amino acid sequence and 3D structure. So, for example, elastase of chymotrypsin. So, they have similar functions such as breakdown proteins but just a different mechanism. Okay? That's why they are what we call as protein families. We actually to do this, to know if these are families, we have to check their amino acid sequence. Okay, so there are so many tools for that. Okay, so bioinformatic tools to do this. Okay, so we can just do this in the internet to check their similarity in the amino acid sequence. Okay. And proteins, so we have multiple peptides, diba? So non-covalent bonds can form interaction between individual polypeptide chains. So for example, katong sa hemoglobin na you have four subunits, diba? You have 2 alpha and 2 beta subunits with iron at the center. So these 4 subunits interact with one another, and ang interaction na ni class have many different ways para mag-interact sila. So we have what we call as a binding site where proteins interact with one another. We also have subunit. Ang subunit is each polypeptide chain. So this is one subunit. So one subunit, one subunit, one subunit, and another subunit. Okay, so kung asa na sila nagbind, kung asa na nagbind ang each, that's what we call as the binding site. Okay? Then ang dimer, like okay, so we have some proteins nga dimer, so it only has 2 subunits. So that means this is called a dimer. Okay? So if you have this class ba, if you have a quaternary protein or dimer, okay, if you have these multiple subunits, so it does not necessarily mean nga same na sila nga polypeptide. Okay, this could be different with this, or all of them can be different. Okay, or all of them can be the same. There is no criteria. Okay. Okay, so for example, for cap protein, so a cap protein is a dimer. So in this case, so this is one subunit, and this is another subunit. The green one is another subunit, and somewhere here is their binding site. So kung asa sila nag-bind, that's what you call the binding site. So in this case, mo ni ang ilang binding site. Okay, so it must be this one. Okay, and then the tetramer. So for example, we have a neuraminidase protein. So this is a tetramer. So one monomer, one subunit, another subunit. Okay, and then mo na ilang binding site. So these two binds here. This is their binding site. Nya kini silang duha. Their binding site is, as you can see from here, it's somewhere here, and then here, and then here. So again, you need a 3D structure to know where exactly is the binding site, but for our purposes of learning, for knowing lang nga naa sila binding site, we can just take a look at this. Okay? So, again, we have different subunit proteins. So, like for example, hemoglobin, it has 2 alpha o 2 beta. So, every subunit plus sa hemoglobin, they are called globin. Okay? So, globin ang tawag ana sa each subunit. And also, we have protein assemblies. So, protein assemblies meaning it's just protein which assembling together, okay, from the word itself. So, they can form long chains if a protein has 2 binding sites. So, for example, this is just one protein, di ba? So, mo ni usa ka subunit. Just one subunit rather. So, this one subunit has this binding site. So, mo ni ang binding site. And if it interacts with another of this subunit, then it could form a dimer, di ba? So, if their binding site are compatible, so of course, these binding sites have to be compatible para mo bind sila. They can't just bind when they see each other, di ba? What if dili compatible ang ilahang binding site? So, the binding sites have to be compatible, of course. That's a requirement. So, if this protein subunit, interacts, because it has a binding site and it interacts with another subunit which has a compatible binding site, then it could form a dimer. Okay? So, another case is that if a protein has 2 binding sites, so it has this binding site, for example, this subunit has 2 binding sites, where this binding site and this binding site. So, it could form multiple because it could form here and it could bind here. Diba? So, it could bind here and then it could bind here because it has 2 subunits. Okay? So, if it could form even this one, so alpha helix na sa diyang structure, and this one is ring na structure, diba? So, it depends on the binding site really. So, if you look at it, yung binding site forms like this, that's what it could form a ring. So, if magsumpay-sumpay na siya, inana dyan siya, diba? Murag ka ng assembled na hula hoop ba? And then we have the actin fibers in muscles and cytoskeleton. It is made from thousands of actin molecules as a helical fiber. Okay, so it's they are just bound together. So that means it contains more than one active site, diba? Because it was able to form this multiple protein assembly. Okay? So, for example, this is just one subunit, then if it forms this like this one, if it forms this structure, it's called the filament. And then if it forms like this structure, ang term lang ana niya. So these are just the terms, okay? Kung ingon ana ang structure nga ma-form. So ang term ani is the virus coat type, and you have this helical tube. Okay? So these are different protein assemblies. So we have 2 types of proteins based on structure, diba? We have globular and we have fibrous proteins. So globular, this is usually again the enzymes. So these are enzymes. Mostly ang globular are enzymes, yun na. So these are compact shaped like a ball with irregular surfaces. Okay, and we have fibrous proteins, and these usually span a long distance in the cell. So, examples of fibrous proteins, you have collagen and elastin. So, these are structural proteins. Okay, we also have keratin in hair. Okay, and then, so this is the structure of collagen and elastin. So, imagine a collagen molecule. Okay, so this is the short section of a collagen fibril. So, kini siya, kini. This one is the short section of a collagen fibril. Diba? This is the collagen molecule. So, if imo nang i-zoom, this is the collagen molecule. And if you zoom this part of this collagen molecule, it looks like this. Okay, so it's a collagen triple helix. Okay, another thing, we have an elastic fiber. Okay, so it's this one, So in any structure, and if you stretch it, then ingon ani gid na structure ika-stretch nimo. But if you relax, so it's like this. Okay. Now to stabilize the cross-links, class, to stabilize the protein, we have we have this disulfide bridges here. So it could form disulfide bonds. So like for example, this could be from serine molecules, CH2SH. So this could be from serine amino acids. So this could form H bonding here. So cross linkages can be between two parts of a protein or between two subunits. Okay, so kini siya, it shows two parts of a protein, diba? So this is just one subunit, diba? This is just one protein, and then this portion of a protein has this SH, and this portion of the protein has another SH. So they could stabilize they could stabilize this protein more due to this presence of the disulfide bond. So that's why it's called intra-chain, meaning sa intra, it's within the same chain. But if ang linkage is from one subunit to another subunit, so different siya nga polypeptide chain, then we have what we call as an interchain. So inter meaning gikan sa one subunit with another subunit. Okay? So we have SH, sorry, this is from cysteine rather, cysteine diserine. It's not serine but cysteine. Okay, so the conformation of a protein gives its unique function. Okay, remember that. Structure determines the function of a protein. So actually, to work, para mo functional ang protein, some proteins must interact with other molecules. Usually, one or a few molecules from the thousands to one protein. Okay? So, ang molecule, so, again, a lot of proteins, class, can't just work if it's just that. This is a protein, okay? So, a lot of proteins are not functional. They can't do their function if it's just a protein itself. So, a lot of these proteins bind to something else. So that something else is what we call as a ligand. So this is a molecule to which a protein can bind. And the binding site is, again, as you know, a binding site is the kanang specific site kung asa na sila magbind. Okay? So this is the part of the protein that interacts. This is a specific part, okay? So it could these binding sites consist of cavity formed by specific arrangement of amino acids. Okay? So, for example, so mo ni ang gitawag nato og binding site. Okay? So, mo ni ang cavity nga na-form due to the specific binding of amino acids. So, this is the binding site. This green one is the protein, and this is the binding site of the protein. So, it binds to the ligand. Okay? So, it could bind to the ligand using non-covalent bonds. Okay? So, for binding of the proteins, a ligand binds to a protein at a binding site. Yes. A protein may have more than one binding site. Yes, you know that. It may bind the same ligand many times, or it may bind different ligands. Okay? And then, ligands can bind to regulate the activity of a protein. Okay, so, it could either activate the protein, or it could either the binding of the ligand could either slow down the function of a protein. Okay? So, it regulates. So, depende sa need sa iyahang function, depende. So, that ligand could either activate or partially deactivate a protein. Okay, so we have also interior amino acids have a say in the conformation of the molecule too. Okay, and so proteins could also pair with other molecules. So for some biologically active molecules are a result of a very cozy relationship. So for example, rhodopsin and opsin. So this retinal, this is a light sensing pigment, and it is bound to an opsin nga protein. Then for heme, that's an Fe, an iron containing molecule, it could bind to a globin protein, di ba? Mo ni sa hemoglobin. Okay, so for the next video, we will have the